Well, I guess you covered all of it, really. I don't really need to say anything else. So, as you said, I'm Josh. I'm representing the Michigan iGEM teams. The title of our talk is Synthetic Biology in the iGEM competition. So the first thing I'm pretty sure a lot of you like to know is, what is synthetic biology, right? Well, I'll cover that first, and then we'll talk about the competitions that we participate in, and then I'll mention what Michigan teams have done, especially this year. So synthetic biology uses much of the same techniques and equipment of the biological sciences, but instead of research and new discoveries, a synthetic biologist looks to co-opt and improve upon genetic blueprints of existing organisms to design and create novel biological devices and systems. So it's more of an engineering field. It's also commonly known as genetic engineering. It's less It's also known as genetic modification. So how it works is, in general, is you have several genes that you like for one reason or another, and you take parts of each gene and you combine them into a composite gene. And you can do this, and you generally do this several times and create several composite genes. These composite genes you insert into a biological system, and you create a synthetic system. The system can be in vitro, it can be in a virus, it can be in bacteria, it can be in yeast, it can be in eukaryotic cells, mammalian cells. Some examples are most penicillin now is produced through synthetic bacteria systems. Recently, there was I don't know if people have seen the Henry Ford commercial where people were able to genetically engineer a patient to cure her cancer. So gene therapy is a form of synthetic biology. They were able to re-engineer her immune system to recognize the cancer. iGEM was founded by some of the founders of synthetic biology. It's as Jim said, it stands for International Genetically Engineered Machine, and mainly runs the competition. The competition involves teams creating open source projects relating to different aspects of synthetic biology. So one of the key aspects of the competition is everything in it is open source. None of it can be patented. All of it has to be submitted for future teams and for the general public to use.
Are there some standards there?
Yes. So that's the other key component of the competition, is that the things that you submit have to meet certain standards, or you have to come up with your own standard. These are called BioBrick standards. So the DNA is actually standardized so that several different teams that follow the same standard are actually compatible with each other, and future teams can actually combine these projects of past teams modularly to create new projects. This competition is an international competition. It was started by MIT in 2004. It's branched off of MIT since then in 2012. It's now its own nonprofit organization. Each competition ends in a fall conference called the Jamboree. And so what is this? So what is this Jamboree? Well, it's kind of your standard conference, except everyone's a student, and they're all undergrads or master's students. I haven't seen any postdocs, but of course there's also advisors, faculty who show up. So there's a presentation. There's my co-presenter Christine and the handsome devil over there. And there's a poster session, and then there's social events. So the competition is actually now split into 3 divisions. There's the high school division, which is strictly for teams associated with high schools. Then there's the undergrad division, which is separated from the overgrad division by whether or not your team has someone over 23 or not. It's kind of an arbitrary breakdown, but it allows sort of even the playing field, or they think it allows it to even the playing field between Europe and the United States, where the education is different and the distinction between master's and undergrad is blurred. So I mean, I actually think that the undergrad team that won this year could have kicked the overgrad team's butt like several times over, but they're trying. They're trying to make it fair. So each division is split into tracks. There is now 15 tracks. These are energy, environment, food and nutrition, foundational, advanced, health and medicine, information processing, manufacturing, new application, art and design, community labs, entrepreneurship, measurement, microfluidics, policy and practices, and software.
Okay.
So these have grown. The number of tracks have grown.
So there's a separate community for every one of those.
It's not necessarily a separate community. It's sort of like a separate. There's separate groupings. So they like to have all the energy presentations together. Like to have all the environment presentations together. Like all the software presentations were all in a row on the same day at the conference.
How many people come?
So this year there was 243 teams.
Yes.
So thousands of people from all over the world.
2,200 or 2,300 attendees in the Heinz Convention Center.
From Boston.
All undergrad or overgrad. That isn't even the high school.
And next year they're
That's rich.
next year they're bringing in the high school students. So
With all this stuff about data, big data, data science, and all that buzzing around, is there a data element to this that's emerged?
well, there's always been the database storing the information of the synthetic parts, the projects from past years, called the registry of parts. So the collection of that data and making that data accessible to future teams is something that they've improved a lot upon and are still working on. It's the software division was one of the earliest tracks. The software track was one of the earliest tracks to emerge in the competition, separate from the normal laboratory tracks. And a lot of the projects that come out of that track deal a lot with handling databases, handling the synthetic biological parts, information and data. As you'll see, the competition is very open-ended. It's sort of like a judged science fair. So the idea is basically they give you some criteria, but it's not incredibly restrictive, and just basically trying to impress the judges at the end of the day.
So what distinguishes the software category from information processing? Is information processing more like genetic computing, or?
Yeah.
Okay.
So information processing track is more basically doing computations, generally speaking, in a biological system.
Okay.
So the Michigan team in the past has actually done very well doing information processing project, creating a biological bit out of DNA. So being able to store and manipulate information in DNA easily was the goal of our project in 2012 and 2013, which is much more stable than information stored in associated proteins, which is how a lot of people currently do it. And we did very well those 2 years. I can go into that a little bit more later.
Thanks.
So at the end of the competition, there's a top 3 for each division. So in Boston, at the end of October, there was top 3 undergrad, top 3 overgrad, and then the high school students at their conference had a top 3. Then there's medals, which are not what you really think normally of as medals. They're sort of like criteria levels. So lots of teams can get bronze, lots of teams can get silver, lots of teams that can get gold. I think it's meant to recognize level of compliance with the philosophy of the organization and achievement, of course. And then so there's also track awards. So the best software project gets an award. The best policy and practices gets an award. The best art and design gets an award. And then there's non-track awards like best poster, best presentation, best new biological part is an award. And so now I'll transition to the bulk of the presentation about what Michigan teams have done in the past. So Michigan joined the competition in 2006. It was very different back then. There was like 5 faculty advisors and a handful of students. It's now pretty much completely student run. We have faculty advisors, but they're really just advisors. We get our funding. We create a budget. We decide how to spend it. We teach each other how to do everything. Students operate at every level of the laboratory and software management.
So does it have a home base here at UMich and one of the engineering, biomedical engineering, or CSC?
So in past, it's mainly been housed in. It's mainly been.
Or MICDE?
It's mainly been primarily sponsored by MCDB in LSNA. Now that we have 2 teams, we're now sponsored by this department and MCDB. So one team is sponsored by MCDB, one team is sponsored by bioinformatics.
And the vice president for research too.
Yeah, but our official sponsor is bioinformatics.
All right, well, you know.
In terms of
whatever, it's beautiful. I think we're making a good investment. Don't you? I mean, this is for real.
So in order to get so the official sponsor status is mainly something that student groups establish with the university in order to use proprietary things like the Block M and to have nonprofit status. So we really just choose one department and fill out the paperwork. I guess you could do multiple departments, but all you really need is one. Past tracks that we've participated in include foundational advance, environment, and new applications. So this year we had two teams. This is the first year we've had 2 teams. There was one team that participated in the health and medicine track, and then our new team participated in the software track. And so the software track is a lot different than the other tracks. No lab work is really required for that track. The goal of the track is really to develop tools for synthetic biology and biological research. So you see projects involving modeling, you'd see projects involving DNA sequence analysis and annotation. You see projects, prediction software that involved in automating the creation of protocols for synthesizing new biological parts. And so I was mainly going to talk about what software we developed this year as our first year software team. So any questions before I delve into that? Okay. I think a little history will help understand our current project. I've actually been in iGEM for quite a few years on the lab team. I've done a lot of lab work and know have a lot of experience with what works and what doesn't work and what really causes struggle. And a lot of times in the lab, it'll seem just like that biology stops working for like a week, 2 weeks, a month.
Welcome to forever. It's not
gonna get any better.
Oh man.
So and that's when really start scrambling for new ways of doing things, and to get things fixed, especially when you have a 1-year deadline for your project. And so what you usually do is you start asking people. You start going to people who do this stuff for years and years and years, and you ask them, how do you usually do this? And sometimes that works. Sometimes you can't replicate what they tell you to do, and when all else fails, you read the manual. Usually every lab procedure, every reagent that you buy comes with a protocol to use, and so you usually try and follow the instructions, and hopefully it works. However, this is not always the case. A lot of the times, the protocols, the procedures that are given to you are for ideal conditions, are basically run through their marketing department to basically cover their asses and prevent people from suing them. So when that doesn't work, we go online, and there's websites like openwetware.org, and go through different forums to try and figure out how other people did this, what problems they had, and what did they do to fix it. But sometimes, even then, you'll encounter a problem that nobody's really solved, or nobody's really shared how they solved it. So occasionally, you'll find protocol-specific websites or pages that will have software associated with them. Like there's one protocol that a lot of people struggle with a lot is called a ligation. And one thing we found when we were having trouble with this was a ligation calculator. This basically reagent calculator was actually created by an iGEM team from Cambridge in 2010, and this is one of the resources we use to try and fix our problems with this specific procedure. So we took all these different attempted solutions, all these different things we've tried to compile them in past lab years. And so this is just a Google spreadsheet that I created to keep track of what I tried for various ligations and what worked and what didn't work. So so things that we that failed just marked off in red. Things that we were currently attempting were in yellow. Things that we just gave up on were in white. And there were supposed to be some things that worked that we'd put in green, but as you can see,
Not at this point. Yeah.
that
There are none.
that was not meant to be.
Boy, this is fascinating, actually. I think many of you know that I don't chair it, thank God, but I sit in on the IT executive committee for the health system. And, every month we have to look at something like this. And I guarantee you that, most of them will be green, and a few of them will be yellow, and maybe one will be red, which is one that they want to kill off anyway. They expect you to believe this. And 60% of IT projects fail. I mean, that's right out of the McKinsey report. And so this looks good.
Yeah,
Just try to get one or two greens in there, man. I mean, but
Yeah.
down at the bottom.
So this is what happened in 2013, and
So, are these all the same restriction enzymes?
the-
Why don't you because you got to keep. There's more information you really need to keep track of because some
Yeah.
restriction enzymes are kind of notorious for being messy.
Yeah.
Some manufacturers are not nearly as good as other ones.
Yeah, my favorite parameter, this is the physical chemist in me, temperature would be an example. I
Yep.
mean,
That use polyethylene glycol sometimes.
Yeah,
Right.
so
Like,
there's,
it can be problematic.
Yeah, it can be probably, it can also work depending on the situation.
yeah, so anyway, we're getting the idea.
So part of the BioBrick standard, part of the synthetic biology standards of iGEM are the restriction enzymes that are used in these these cloning operations. A lot of these enzymes were picked originally for their efficacy and for their specific properties like temperature sensitivity and things like that. And
I
so
don't want to take you off track, but there's a lot of things are very batch to batch. But anyhow,
Yeah. So some of those variables have been reduced by the BioBrick standards. Is what I was getting at.
they actually have a repository. You order the restriction enzymes from?
You order the DNA.
Okay.
So as part of your team registration fee, you get access to all the submitted DNA parts from all of time. And so you just put in a request for a certain DNA sequence and they send it to you within a week. And that's basically paid for by your registration fee.
When you order the enzymes, usually from a company.
Yeah.
Is everybody in it from the same company?
Hard to say.
That's what
I think Jeff's making his point.
Jeff needs to be a judge here.
A lot of people order from NCBI. Sorry, NEB. Sorry.
Yeah, New England Biomes.
They're good for restriction.
Yeah, they're good. Yeah.
For
That
Anyhow,
so actually,
I don't want to take you off.
So actually,
we
no,
want to let you
this
get
has-
off this.
this has exactly to do with what the software that we came up with. Anyways, in 2013 we had this issue, and a lot of us, including myself, just gave up. We just stopped doing work. Nothing was working. We couldn't continue, and we just had to basically settle for what we had at that point. So this past year, what I and some others decided to do was form a software team and trying to just address this problem from using a more advanced approach than what you saw previously, which is basically just a spreadsheet. So some of you might know Basil, he's in your department. A couple of us were from the lab team in the previous year, and then we recruited a few others from a variety of different backgrounds to come together and learn to do software, learn how to program, how to create HTML interfaces, and try to address this issue. So one thing we did
Oh, wow.
at the beginning of our project is we started looking in the literature for how much this problem had been studied already. And one of the papers we found was this in PeerJ, it's an online journal, studying some of the problems with reproducing known protocols that were published in well-known papers.
Marcy, make sure to send this around to everybody, this reference.
Yeah, I can
Good.
I can share this.
This is
PowerPoint.
we all need to pay attention to this one.
Yeah, Josh, if you can send me the PowerPoints, I can
Okay.
pass them out.
Yeah, good, thanks.
And so one of the things that it found was that almost 50% of the time, the results from these well-established papers were not reproducible. And these are in well-known papers like Cell, Nature, Science.
In
So-
fact, those are the worst.
Yeah.
Those are the worst, yeah.
Was it really the worst?
Yeah,
Yeah, well, see, this is, when it started,
you're
this
right.
was kind of focused on more clinical research. You know, Amgen and J&J did some internal studies, and then that got out. And then, you know, now we're looking at more broadly into the basic sciences. And, you know, the numbers, Nature, Cell, Science, you know, in the J&J Amgen studies were approaching 75% of the studies could not be reproduced.
Yeah.
And so, yeah, there's been big time conferences at the NIH on this one at the director's level.
And I think it's a big threat to, I think, just the credibility of science in
This
general.
is this has the potential to undermine, you know, how we do, you know, promotion, tenure, you know, trying to figure out, you know, and there could be some neat studies that you could do is, you know, somebody publishes a result, some famous lab publishes a result, and all these other labs glom onto it, and you could make a citation tree, and you know, so there, you know, there's that kind of thing too.
Yeah. And I think maybe one of the reasons why Nature, Science, Cell are so bad is because people don't bother to check because they think it's Nature, it's Cell. Like, I don't need to check that.
There's been some other things written about this, the whole hyper competition thing, for an example. You know, the pressure on all, you know, from grad students on up to full professors to keep their CVs current. We've been, you know, everything's being made visible on the internet now. I mean, we've been struggling this for the faculty. Turns out that our CVs are all having to be encoded into NCBI right now format and uploaded on NCBI and linked out to ORCID. You know, it's a different game. So this is great that you're bringing this up, Josh.
Yeah, and one of the things that it found was in several cases the actual the specifics of what a paper actually did of their protocol was actually completely indecipherable. So could not be identified at all. And so what you see here are different aspects of protocols in the biological sciences. A lot of protocols use antibodies or specific cell line or construct, knockdown reagent, specific organism. And in different biological fields, this is how often you could actually identify the correct one. On average, it's barely over 50%.
Yeah, it's getting there's that magic number in there, which is random distribution.
Yeah, I mean, in some things it's worse than others, but it's still pretty dismal, especially for what's supposed to be a hard science.
Interesting how that cell line stuff is bouncing around, and it's pretty understandable why that would be the case.
Yeah, and you think like immunology would be better at its antibody reporting, but no.
Cell lines really well.
So this problem has been established before,
Yep.
And what we hypothesize is that this problem is caused by a combination of factors involving poor documentations, like you saw in the previous slide, difficult to find or difficult to access relevant information, and lack of validation, sort of lack of peer review, lack of replication.
Right, and the documentation is on which version of the software, what data, where, the whole line.
Mm-hmm.
So yeah, so this does play into the data science thing pretty well, this one.
And
Yeah.
I think so does the solution. So we decided to do some part of the competition is an emphasis on outreach. It's an emphasis on what they call human practices or policy and practices. And so there's actually an award for that, and we did. Well, actually, just for our own purposes, we decided to reach out to the scientific community, to professional labs, to industrial labs, to other iGEM teams to find out what their experience with this problem was. So we sent out a survey to undergraduates, graduate students, professional scientists. We have one software guy in there, and I think we got so far we were able to get around 60 responses. The survey is still on our iGEM website for people to take. So we're hoping to get more survey results. We asked how often they were having problems with replication, and nobody was perfect. Nobody had a clean record on this, and most people had problems over 25% of the time, up to 50% of the time, and a significant proportion had a problem the vast majority of the time. We wanted more detail as to exactly what was causing this problem, and so we asked what people were having trouble with. Just lack of specificity and clarity was a major one, or just complete omissions were the main problem. These results are also available on our iGEM website. So we also got feedback as to what people would like to see in a solution. We offered several suggestions, and we got some more beyond that. But mainly, people wanted to be able to compile and browse through a collection of protocols which they could contribute to, and most importantly, be able to review and comment and answer and give feedback for their own protocols, for other people's protocols.
So is that the number of respondents is 66, is 100%? I mean, down on the bottom. I mean, how many people actually?
Oh yeah. Down at the bottom is the number of people who wanted this feature. So people could select multiple features, and the most commonly selected one was be able to browse. And so the specific question we asked was, what features would you use? Some people didn't feel like they would use a certain feature, they wouldn't select that one in the survey. So we also, using this information, we set out to create a solution, and we decided to call that solution ProtoCat. So what is ProtoCat? ProtoCat is a protocol catalog. That's what it stands for. And the idea is to crowdsource a registry and review system for lab procedures. And what we figured was on our own, we were only able to create this spreadsheet with a limited number of trials. And we did lots of trials, but it wasn't enough. So we needed more manpower. and we figured the best way to do that was to crowdsource the problem. And so we set out to do this we could, by considering several different programming languages, several different several different packages for those languages, and we settled on a system where we would put ProtoCat on a server, which the code for which is on GitHub, and scientists and journal readers would access the entries on the server and be able to collect all the information in one central location. So some software has people maintain their own basically version of databases on their own machine, such as like the annotation software called Ape. But in order to eliminate conflict and confusion between people's different versions, we wanted to keep it all in one central location. And so we thought the best way to do that was using a Python package called Django. It was recommended to us, and we decided that we'd all learn this language, learn Python, and learn how to use this specific package. Of course, lots of us came from different programming backgrounds. Some of us knew Python, some of us didn't. So there was a lot of learning that went into this. What Django is it's a Python library. This library simplifies the generation of web pages from a database, and it allowed first-time programmers like a lot of us to do this more easily, more simply. Since it's open source, it would allow future programmers to add on to the software more easily. So where did we get to at this point? This is the home page that of the current version of ProtoCat. There's also like the catalog and an account system that we set up. And of course, there's like our contact information and whatnot. It's got a pretty simple, basic entry method at this point. Basically, it's the protocol is just contained in a text box for now. Once you enter the protocol, you can browse the existing ones in a list, and you can select the protocol and then read its contents. Like a basic web page. At the bottom of every protocol, the owner can delete it or post a comment. Well, no, anyone can post a comment. And every entry has its own web page, so you can link to it from other sites. So when we started out with this project, we had a lot of ideas for things we wanted to implement. And so we have a lot of ideas for what we want to implement in the future. If we have the chance to work on this in the future, want to implement.
Do you think deleting is a good idea?
Yes.
I'm not sure I do.
Because
it tracks how things have changed.
Exactly.
There
And sometimes
may
you
be
have to want to go back.
so if you, well, for now,
There's information in there that might be what didn't work.
Yeah.
That's right.
So one thing that was suggested that we definitely want to implement is some kind of version control, so that people can edit. So the deletion system is currently what's allowing people to edit the mistakes they make when they enter something. Being able to go back and see how a protocol has improved, or how what someone said was the right way to do things and are now saying something different. So in terms of that, I agree. Currently, that's the main method of correcting protocols, but that has to be updated.
Yeah, just to follow up on that, I mean, considering that the purpose of this is for replication, if someone were to do something and refer to the protocol on a particular page, and then someone goes back and edits it or deletes it, it's no longer the same protocol. So I think the versioning is that sort of system is almost paramount for this to be effective, just because you need to be able to trace back and say, well, we use this protocol. If you use the exact same protocol, then you'll be able to reproduce the results.
Yeah, absolutely. And so we've been thinking hard about how to implement that kind of version control, and we're
It's
still
not
not
simple.
still not sure. So
Well, actually, if you want to implement the version control here, probably you want to think of using some Python wiki module, because most of them have a pretty, I would say, mature system to do like text-based version control. And most of them also have like a talk page similar to what we have in Wikipedia, which can serve the same purpose for you as the current comment system.
we'll definitely keep that in mind. We'll definitely keep that.
Yeah,
in
that's a good idea.
So I have a question. So I'd like to challenge one of your assumptions here. What if in these unreplicatable protocols, there's a variable that the person is not aware of? For example, simple things like temperature, or that their distilled water somehow has
Something else in it.
Yeah, something else in it, like iron or something,
Been there,
that causes it
done
to
that.
work or not.
Yeah.
And so, how do you deal with that? I mean,
So what we're thinking is that for variables that people miss, we were hoping that the review system would be able to catch things like that, and people who figured out that there was that variable would comment and say, you got to take this into account and make a new version of this protocol. From the outset, the idea starting from last year was to try and keep track of as many variables as we could, and try and keep track of which values for each parameter worked, which ones were just not important, etc.
right.
And so forth. And so we'd like to implement a sophisticated search function, a rating system, so that people can sort protocols by, it can filter out the bad ones from the good ones without going through all the comments. Other types of filters, like by category, would also be nice. We also don't think the current input system is adequate at all. We'd like to have something more sophisticated, where maybe the protocol is broken into entry of steps, and maybe even have separate entry of the amount of time each step takes, and just a more sophisticated breakdown of what exactly a protocol is would be nice to see. We also like to people to be able to look at the users and be able to maybe even review the users and say, like, this person is really good, or this person is just filling out space on this database and ignore everything they say, stuff like that. So similar to what you said before, we'd like to. One of the reasons why we chose Python was so that we could implement calculations within the protocols. I think what we accomplished this year and what we set out to do was create a foundation for all these advanced features. Although what we have at this point is pretty basic, what it has built the foundation for can be very advanced. And so we'd like to, for variables like temperature, like concentration, like volume, like buffer buffer concentration, things like that. We'd like to allow for variations between different people, between different instances of the protocols, so that in the same protocol page you could say, I want a liter of product. I want a 5 molar concentration of product, and it would tell you, okay, you need to add this, and this amounts in order to get this product, instead of people having to go to several different pages to see the 1 liter version, the 2 liter version, the 5 microliter version. And so that was one of our main ideas from the beginning. One cool idea we also had was actually suggested to us from our lab team, was to have sort of a timing system, sort of like a cooking show, where you could push play, and it would tell you at this time, okay, you need to go back and start this next step right now, or else your gel's going to run off, or something like that. So sort of making protocols more idiot-proof would.
Text reminders on your phone, huh?
Yeah, exactly. Like, don't forget to
Right.
take that out of the oven.
Yeah.
Stuff like that.
Explode.
Probably it's better to implement that as an app instead of putting it on the website.
Sure.
Yeah.
Yeah, you can close your browser, but you cannot turn off your cell phone.
Oh, yes.
Right.
I
Oh, you
can. I'm older. I can turn my cell phone
Cell phone has no app.
Or if you still have a beeper, maybe send you a page.
Yeah, there.
And so right now, right now it's currently hosted locally, just on a development server, and we wanted to get it on a global server by the end of the year, but we ran into some difficulty with that. So we'd like to, one of the first things we'd like to do next is to get it on a global server, and get it hosted on a permanent global server, maybe even by iGEM itself. We'd also, even though we want this to be centrally located, in case of people want to work offline, we'd like to be able to give them the option of downloading multiple protocols, maybe in the entire database. Especially if they don't want to have everyone, if they want to make changes, don't want everybody to see the changes that they make while they're still working on it. We'd also like to have like vendor information, so like if it tells you need use this much of each reagent, they can have the information to go and buy those reagents, and maybe even be able to purchase them directly through the software. We're hoping we can actually get sponsorship through that means as well. Maybe have like little ads within it. That's how we'll pay for it. And so, in order for this whole thing to work, we get a protocol sharing peer review community has to be cultivated. If it's just a few people on our iGEM team doing this, it's pointless. Just like the iGEM competition itself, it only really has an effect if everyone actually wants to use it, actually uses it, actually contributes to it, and gives feedback. In terms of the future of our team and our club, we'd definitely like to increase our survey size, because 60 is maybe fine for a first attempt, but it's clearly not enough for like any sort of scientific result. We'd also like to send out a beta version to the people who took the survey. One of the questions is to sign up on the survey is to sign up for a beta test. For next year's team, we'd definitely like to recruit more talent. So if anyone is a student who wants to work on this, or wants to work on an iGEM project in next year, please come talk to me or Jim, and we can definitely get you involved. If you're a faculty or staff, we'd love to have you as an advisor. We can use as much help as we can get. And of course, we want ProtoCat to win iGEM. We definitely want to bring recognition to the university and all the hard work that we've done and all the support we've gotten from departments. so far. So we'd like to thank our supporters, our sponsors, mainly you guys, the Bioinformatics Department, and ARC. We really appreciate
Office of Advanced Research Computing.
Yeah.
Yeah.
We
Well,
really appreciate all the support you guys have given us this year.
well, we want to figure out how we can do more to help sustain this effort.
Yeah.
I think is really our goal. And we're partnering a lot with ARC on a lot of things. The guy who runs that, Eric Mickelson, is terrific. He meets with me every week, every Monday morning. So there's some ways to build from this, you know?
Yeah, of course we want to thank Jim and our other advisor, Mark, for all their advice and their support.
I'm sure you'll be able to, this is a fantastic start, really. And thank you, Jim, for bringing this to us. And, I know that there's faculty sitting right here, I know, would be willing to help.
And thank you all for listening.
Oh, wow, look at that. I was driving down Washington the other day with a visitor coming home into work. And there was some half-naked guy with a wheelbarrow and a bunch of paint. A bunch of paint cans like in this wheelbarrow. Maybe it was you, Jerry. I mean, somebody want to know what the hell is going on. I mean, they were like wondering, what is that? Yeah. Well, that's what it is. You guys are painting rocks, taking over the rock.
Yeah,
Got a couple questions.
that's fun.
No, it's great.
A couple questions. So, at the very beginning, you mentioned they have a track for alternate design.
Yes, that was
What
new
was
this
that?
year.
The alternate design track in the iGEM competition.
Yeah.
What was that about?
So that was new this year. It was a lot about exploring the appeal and beauty of that can be created through synthetic biology. And the winner was I believe a team from France. I think it was a university in Paris that created an artificial lemon, and it was supposed to taste and smell like a real lemon, but
It was a little lemony.
Another question is,
Still taste it.
I actually noticed that your presentation pretty focused on the web lab part. I look at the interface, I didn't see any problem to extend the web to dry lab protocols.
try that.
Yeah, because actually, other than the web lab published journal, they also have some like Nature Cell journal that do like dry lab computational analysis, and they don't follow like traditional protocol.
yeah, that's
got
for sure.
criticized.
Yeah. So one of the ideas I didn't mention was to sort of have the modeling of the protocol's results also attached to each protocol. So maybe just even if it's just like a video at the end of the protocol that shows either simulates or shows what the expected results should look like. That would be a good way to share and review modeling. In terms of sharing software, I think that would be a little harder.
But it's not necessarily software.
For
right?
files. Yeah.
It can be parameters files, like
Yeah.
you do
Yep.
proteomics analysis.
Mhm.
Yep.
You say, like I set a false discovery rate at 0.01.
Mhm, mhm.
They say like 0.05, and
Yeah.
they can have some problems there.
I've actually led the modeling on the lab team, the math modeling on the lab team for the past few years, and I'm definitely interested in how we can integrate that. If not in something more advanced than just the comment system.
Yeah.
It would be nice if maybe when you enter your parameters into a given protocol, it could predict certain parameters. It would also be nice if like people could report if there was an easy way for people to report the results of their protocol in a standardized format. So like they could enter like, I did this protocol and this is the data from my results. And they could share that and people could compare and maybe compare the calculated parameters that, from the different results. So we're definitely definitely thinking more about how we can expand to that. So that's a good idea.
You guys looked at electronic lab notebooks. You know, we have one that we use in our lab called LabKey. It's in the open source. It's really pretty good, and it might be a way to, you know, allow folks to, you know, in their own lab environments, you know, have something digital. Then could be probably pretty easy to aggregate up and share. You have a certain part of it be viewed by. I think we need them.
Yeah. As long as something like that is public.
It is, you know, we went through that. I mean, this is, you could, Alex, you're our expert, but I think you could, it's in the open source and you could configure it to expose it to the public.
Because I think that one of the big contributors to this is that people figure out how to fix these problems, but they just write it in their lab notebooks and nobody.
Exactly, so I'm trying to get that out into the digital lab notebook.
So I know you showed a couple statistics on this before, but how receptive were the people you reached out to towards the idea of sharing like their specific labs protocols for for whatever field? I mean, because it seems like, especially for the most bleeding edge stuff and like the hyper competitive areas, it seems like everybody's trying to compete against each other, especially in terms of what you talked about with like the Nature, Cell, and Science papers and stuff. They're trying to scoop everybody else. So how do you foster an environment where even those labs in those hyper-competitive areas are?
People we surveyed, the second most popular feature was being able to submit their protocols.
Mm-hmm.
So people want to share. One thing I didn't mention was that the people that we talked to at the conference were very enthusiastic about this. Very enthusiastic about being able to use a system like this, to be able to get help with what they were struggling with and get feedback on their own protocols.
I think there's also a difference in at the iGEM conference, everything is open source, so everybody's about sharing and agreeing on these standards so that my brick can be used in your project next year. It's much more of an engineering model than it is a reclusive private lab view of things. But I think to that point, though, as we get more and more into this, reproducibility is an effective publication criteria.
Mm-hmm.
Then there's going to have to be places where you put your protocols so that they can be vetted and scored so that your data is reproducible. I don't think it's going to be quite as easy. I don't think scientists are going to run to it.
Mm-hmm.
But I think depending on the mandate of the funding agencies and of the peer review process, that may force people, and then you'll have to have a system in place to do that. I mean, I don't think you'll change that culture overnight, because some of those people are ingrained in, I must keep my data secret so I don't get scooped, so I get my paper. It's part of the culture we've rewarded.
So, I mean, this is actually even a problem within iGEM. There's not all teams completely adopt the open source iGEM philosophy. There are a lot of teams who will say in the presentation, I can't talk further about this because a patent is pending.
Yeah.
And
I know that one.
There's even teams that have completely quit, who have refused to report at all anything because they instead wanted to patent their project. So that is still that's not a problem that iGEM is free of.
Even the founders have a couple of spin-off companies. I mean, there has to be something that supports those kind of entrepreneurial endeavors. But great. Anything else, Alex?
Just to follow on to the dry lab and the electronic lab notebook comments and something to think about. One of the things that we've started using for software is these pipeline frameworks, like Taverna or Loney, where we set up a pipeline of tools and input the parameters, and then the pipeline saves all of that information, all the versions of everything, all the input values that we had. And then we just share those files with each other, so we know what versions of everything each other used and what inputs each other used to run that pipeline.
Yeah, that might be something to add. Something like that we can look into.
You present in KCRM before.
Yeah. The best part about making this an open source, crowdsourced project is that people who want to implement features can go on to GitHub and download the software and then implement these features and publish a better version. So we're hoping that not only will we get a user community, but also a developer community going.
From from a librarian, the librarian perspective, in terms of, I think it's probably useful for you guys to look at the existing sort of commercial products and protocols, and sort of what features and metadata it is that they're having filters, because a lot of people are used to looking at protocols that way. So I think it would increase your chance of adoptability, as well as it might be useful to have the librarian like Marcy or somebody to work, especially on something that's a catalog of things that's openly available. Just because there are some other heuristics, and one thing I would say is, if you make it easily citable, it will also increase its adoptability.
Yeah.
Like if you make a protocol that much, it's easy for me to just, and I can put in my paper and done.
Yeah.
Then you'll just make it that much more adoptable than just any
No, that's a good
point.
That's good.
So
good
it may be
practical
worth,
advice.
in terms of usability issue, you might have talk to Marcy, every once in a while.
That was the main reason. That was basically the main
I'm
reason why-
voluntolding Marcy, but all I'm saying is those aspects of it, right? If you're looking to create the crowdsource, right, you need to make it easy to use, and that's where we could say we'd have a lot of experience.
Yeah, that was the main reason behind why we made sure to implement the URL for each protocol. And when I was at I actually presented this poster at the ARC symposium.
Yeah, just
And
recently.
and a librarian, I got in touch. A librarian came up and I got in touch with her about the current protocol commercial protocol databases that are currently being used. So we're definitely in touch with those people, and we have those contacts for our future development.
It's not necessarily just URL. You can provide some functionality for exporting BibTeX or EndNote.
Yeah, that'd be great. That'd be great.
Yeah.
Marcy
will tell you. Mendeley, I don't know, one of them. Someday it'll all work.
It's already on.
It's our dream.
How is it
If only they
licensed
worked.
so far?
It's in terms of license, it's all public. Yeah, it's all public domain, open source. That's as is required by iGEM. Our GitHub group is MIBIOSOFT. Thank you guys for. Nice job. Thank you.
Thank you for your